Welcome back to another Explainer. Today, we're diving into one of the most infamous biological crimes in human history. We are talking about a microscopic mystery, a literal cellular heist that started with a sudden, truly terrifying medical anomaly back in 1981. Imagine a burglar so sophisticated, it doesn't just pick the lock to your house, it actually rewrites the architectural blueprints once it gets inside. Well, that is exactly the story of the human immunodeficiency virus, or HIV. Here's the roadmap for our investigation today. We'll be covering 1. A sudden medical mystery. 2. The anatomy of a break-in. 3. Hijacking the host cell. 4. The immune system collapse. And finally 5. How medicine fights back. Let's kick things off with Section 1, a sudden medical mystery.
To really appreciate this, we have to rewind the timeline a bit. Historically, notorious infectious diseases can usually be traced back centuries, sometimes thousands of years, through ancient texts. But HIV was a completely different beast. Before the 20th century, a highly similar virus called SIV was totally restricted to chimpanzees. It wasn't until the 20th century that the virus actually made the jump from monkeys to humans. And then, just seemingly out of nowhere in 1981, the CDC reported 5 cases of an incredibly rare infection called pneumocystis pneumonia in previously healthy young guys in California. Unlike ancient diseases that we've battled for millennia, this was a sudden, ferocious terror that seemingly emerged overnight.
Okay, moving right along to Section 2, Anatomy of a Break-in.
To understand this break-in, you really have to look at how completely backward this virus operates. In standard biology, your DNA carries your genetic code, right? And that gets transcribed into RNA to act as a messenger and actually get things done in the cell. But HIV, it does the exact opposite. It's a retrovirus. It comes in as an RNA virus, but it transcribes its RNA strand backward into DNA. It's an entirely reverse process from what our cells are used to, which is exactly how it manages to fly totally under the radar.
So how does this infiltration actually start? Well, when the virus enters the body, usually through the skin or mucous membranes, it doesn't even hit its primary target right away. Instead, it bumps into our macrophages. The virus infects a macrophage first, and this is where the heist gets ridiculously clever. The macrophage, just doing its normal day-to-day job, basically acts as a taxi. It dutifully carries the viral invader straight into the nearest lymph node, which just so happens to be exactly where millions of the virus's primary targets live, the CD4 lymphocytes, or T helper cells.
So how exactly does HIV pick that cellular lock? Most viruses use a single adhesion molecule to grab onto a host cell, but HIV uses 3. First, a viral molecule called GP120 grabs onto the host cell's CD4 receptor. Then, GP120 literally changes its own shape to bind to a second receptor, either CCR5 or CXCR4. And here is the profound irony of the whole thing. Those secondary receptors are chemoreceptors. Their normal job is to receive chemical signals that warn the cell about a microbial predator. HIV ironically uses the cell's very own alarm system as a literal doorknob to sneak inside. Once both are grabbed, a third molecule, GP41, activates, harpooning the cell membrane and pulling the virus inside. Just like that, the burglar's in.
Which brings us to Section 3, Hijacking the host cell.
Remember that backward RNA to DNA trick we talked about earlier? Well, once the viral RNA is copied into a double strand of viral DNA, it could ship directly into the host cell's nucleus. Here, the virus drops a highly specialized enzyme appropriately called integrase. Integrase physically splices that newly formed viral DNA directly into a host cell chromosome, and it specifically targets a section of the chromosome that is active. At this point, the host cell has been completely, 100% hijacked. It reads the viral DNA just like its own code, turning a once healthy cell into a relentless virus manufacturing factory.
As this newly hijacked factory starts churning out viral parts, it builds them in long, continuous chains called polyproteins. But before the millions of new viruses can assemble and break out, they need one final, crucial assembly line worker. Enter an enzyme called protease. Protease acts like a microscopic pair of precise chemical scissors, snipping those long chains of proteins into individual functional pieces at exactly the right spots. Once they're snipped, these pieces assemble into complete new variants, ready to burst out and seek brand new cells.
And that sets up Section 4, the immune system collapse.
The host cell isn't completely blind, With all this viral DNA and foreign machinery floating around inside, the cell's internal alarm bells eventually trip, activating a chemical called caspase-1, and this triggers a specific, violent type of cellular self-destruction called pyroptosis. Think about that word, pyro, meaning fire. It is literally a fiery, explosive event. The infected cell suddenly and violently bursts, releasing not only the viral proteins but also massive amounts of inflammatory cytokines.
But here is the absolutely tragic paradox of that explosion. That fiery death, it's supposed to be a warning flare. It's trying to call in the immune cavalry to help. But instead, it draws thousands of healthy CD4 helper T cells straight into a toxic, highly inflammatory war zone. Because of this, a really unusual feature of advanced HIV is that exponentially more CD4 cells die as collateral damage from this friendly fire alarm response than are actually infected by the virus itself. Isn't that wild? This mass casualty event among the immune cells leads us to a highly critical number, 200. Normally, a healthy person has somewhere between 500 to 1,200 CD4 lymphocytes per cubic millimeter of blood. But as this microscopic heist and the resulting fiery explosions continue unchecked, that number just plummets. If a patient's CD4 count falls below 200, their primary line of defense is fundamentally broken. They become fully immunosuppressed. And when those vital CD4 helper cells are gone, the body entirely loses its ability to contain other common pathogens that live inside our cells. Without the CD4 guards there to hold them back, these internal infections just run absolutely rampant. We're talking about everything from parasitic toxoplasmosis to fungal pneumocystis, viral Kaposi sarcoma, and bacterial mycobacteriae. When a previously healthy person begins developing these formerly rare opportunistic infections, that is when the diagnosis becomes Acquired Immune Deficiency Syndrome, or AIDS. The immune system has essentially collapsed.
But hey, the story definitely doesn't end there. Let's look at Section 5, how medicine fights back.
Back in the early 1990s, therapies relied on single drugs, and it just wasn't enough. Doctors quickly realized that to stop such a complex break-in, you need a highly coordinated defense. Enter HAART, or high-activity antiretroviral therapy. HAART uses a combination of different drugs simultaneously to completely corner the virus. It's an incredibly elegant and aggressive defense mechanism that has completely transformed the fight against this disease. This coordinated defense builds perfectly on everything we just learned. These medicinal roadblocks target the exact stages of the break-in. You have drugs that block the initial attachment by targeting CCR5 and GP41. You have drugs that physically stop the viral RNA from elongating into DNA. You have anti-integrates drugs that prevent that hostile takeover of the cell's nucleus. And finally, you have protease inhibitors that stop those chemical scissors from assembling new virions. By hitting all these choke points at the exact same time, the therapy severely cripples the virus's ability to replicate. Now, you might be wondering, why is it so absolutely crucial to use all these drugs at exactly the same time? Well, it's because the viral machinery is incredibly sloppy. When HIV copies its RNA, it makes a ton of mistakes, resulting in constant mutations. The source material actually points out that HIV is notorious for these changes, which can enable it to evade single drugs. But with combination therapy, if the virus mutates and gets around one drug, the chances are extremely high that it's still going to get trapped by the other 2 or 3 drugs targeting entirely different mechanisms. Of course, running a security system this complex requires a serious command center, and for this, that's the clinical lab. Through scrupulous diligence, doctors constantly monitor a patient's viral load, which is the amount of virus present, and their exact CD4 lymphocyte counts to ensure the HAART combination is working. Thanks to this rigorous monitoring and combination therapy, HIV is no longer the automatic death sentence it was in the 1980s. The incidence of full-blown AIDS has dropped precipitously when this therapy is followed, turning what used to be a terrifying scourge into a highly manageable chronic condition. Which leaves us with a pretty profound realization. By understanding the exact step-by-step mechanics of how a microscopic burglar operates, from its reverse genetics to its tragic exploitation of our body's own alarm system, science was able to build a molecular wall to stop it. As we continue to manage this ever-mutating invader, it really makes you wonder, what other seemingly impossible medical mysteries are out there right now, just waiting for the right combination of scientific diligence to unlock them? Think about that until our next explainer. Keep learning.